Thank you very much. So hope this comes out clear. Yeah, thank you very much for inviting me here to Toulouse and this kind introduction, Andreas. It's amazing here. It's really beautiful. It's good to see so many plant scientists. So I am the outsider, I realize, and yeah, it's a little sort of nerve-wracking to say that I'm moving into plant science, but definitely come from a different background. So I was in Toulouse the first time, I think, 23 years ago. That was when I just finished high school, and I had this year where I was trying to figure out what I wanted to do. So I lived close to Albi and was biking a lot around there and reading a lot. And then I stumbled on these Danish books about a traveler and a sailor. So I was like, okay, I think I'll study biology. I was always interested in this. So I moved back to Aarhus. So Aarhus is positioned on the mainland. So it's Copenhagen would be over here on the island, but I moved back to Aarhus and started studying biology. So the campus in Aarhus is here. So this is this green campus with these yellow brick buildings and the old city center that dates back to the Viking Age. But it's really on the bay, and it's positioned in a nice place. So this is where I started studying biology. I thought I wanted to be a marine biologist. I never was, but I ended up working. I basically found out that I was interested in sort of the molecular mechanisms. That was really what was driving me. So I started working on the ribosome and worked with Vinky Ramakrishnan at the MRC in Cambridge on this, and then later moved to Boston and worked on the nuclear pore complex. But now I returned back and I'm in this building. So this is the old hospitals that was bought by the university, and now this is where we can expand. So 2 years ago, we set up the lab over here in this University City 2.0. So this is my group, and we all now work on plants with the question that, so basically land plants, they started to colonize the earth 450 million years ago or so. And many plants, they needed, of course, this extensive root system and developing of this root system. And that has enabled through these many years that plant roots and the microbes that are in the soil, they are in close contact, and they of course co-evolved. And some really remarkable things has happened during this co-evolution. And we are interested in sort of the molecular events that enabled some of these close associations. So in particular, what I will talk about today is the molecular innovations that enabled the transition from a very old symbiosis called a mycorrhizal symbiosis into, well, a more recent symbiosis that's called root nodule symbiosis that only occurs in a few plants. So if we go back in time, these 450 million years ago, where plants start to colonize the earth, very early on, we have this symbiosis with the buscular mycorrhizal symbiosis, and it's maintained in all of these plants today. So this is really a tight association where the fungus invades the roots and colonizes individual cells and makes these symbiotic organs called abuscules, which are really organs of nutrients exchange. So the plant gets phosphate, and in return for assimilated carbon in the form of sugars and phosphates. So this was established really early on, and there's an old signaling pathway in place to control this on the plant side. So there's a very ancient signaling pathway that relies on the fungus giving out signals. One of these important signals are what we call the MYC factor or mycorrhization factor, which is really a chitin, so an N-acetylglucosamine that's decorated. And one of the important decorations is this fatty acid at the non-reducing end. This is perceived by plant cell surface receptors, certain types that are called lysine receptors, and they have an intracellular kinase. So this is perceived. And very early on, we see symbiotic signal in the form, for instance, of calcium spiking around the nucleus. And this is decoded into into the symbiotic response. If we then fast forward 350 million years from this event, then around 100 million years ago in dicot, something happened. So this was a transition of this old symbiosis into a more recent one that we call root nodule symbiosis, where rhizobia or bacteria are accommodated intracellularly, and here they can fix atmospheric nitrogen in return for carbon. So we are very interested in what happened exactly here. This gave rise to this, what we call the nitrogen fixing clade, and plants that can fix nitrogen. So the interesting thing is that this old signaling pathway was something happened to this to enable this new symbiosis. So instead of having symbiotic fungi, it was symbiotic bacteria, and they produced a molecule that was very similar to MYC factors, but we now call these NOD factors for nodulation factors. But the establishment of this new symbiosis is dependent on the same pathway. They don't control opuscles, but they control two processes. One is organogenesis, where we get these formation of root nodules, and then the control of colonization of these by bacteria in a process called infection. So we wanted to understand what exactly happened here that enables this transition from mycorrhizal symbiosis to root nodule symbiosis. So it turns out that in this nitrogen fixing clade, there are both plants that do have this trait, but there's also plants that does not have this trait. And some landmark papers in 2018. So this is one group led by Pierre-Marc and his team, and another group led by René Gertz and his team in Wageningen. They basically sequenced a lot of these plants in the nitrogen fixing clade and compared plants that had the trait and plants that did not have the trait. And what they found was that there's 3 genes that are consistently lost in those that lost the trait. So it's a transcription factor called Lynn. It's a protein called RPG, and it's receptors important for NOD factor perception. So the take home from these great papers was really that there was a single gain here and multiple losses. And importantly, AM was maintained in those species that lost nodulation. So saying that these 3 genes are really important for this transition going from mycorrhizal symbiosis into root nodule symbiosis. So my group has, in the last 8 years or so, tried to understand how NOD factors or these symbiotic signals, how they are perceived. So we know that this is now in lotus or in legumes, we have 2 receptors, one called NFR1, one called LFR5, that are absolutely essential for this perception. But we wanted to understand how is a NOD factor actually specifically perceived. So NOD factors, they are these decorated chitin molecules with decorations that tells the plant which species of bacteria they come from, and then on the reducing end, they have this fatty acid chain. So we could purify a NOD factor, and we could purify the ectodomains of this receptor and show biochemically that these 2 molecules interact. So the receptor binds the NOD factor with a certain binding kinetics and a certain specificity. So we could really show that NOD factors are specifically recognized. And by having mutants that lack any of these decorations and purifying these, we could see that really abolished binding. So it's important for the NOD factor receptors that all these decorations are there. So a NOD factor receptor is specific, and all decorations are really monitored. Another hallmark of legumes that sits in this nitrogen-fixing clade is that they're able to respond and to bind this nut factor and respond to it at very low concentrations. So there's really a high stringency in this recognition. So what I'm showing you here is a concentration series of different ligands that are chitinous ligands or very close to nut factors. And then the readout is this symbiotic calcium spiking in the nucleus. And what you can see, and what is really a hallmark for legumes, is that they're able to respond at very low concentrations to the cognate ligand and distinguish them from these other ligands over here. So this is really a trait that seems to be very important for this transition from AM to root nodule symbiosis, that nut factors are specifically and recognized at a very high stringency. So to understand the evolution of stringent nut factor perception as a key innovation, we tried to see if we could understand this from one of these receptors called NFR5. So we purified the ectodomain so we could express this in insect cells and we could crystallize this. So this is the work of a postdoc in the lab, Kira. And even though these are quite tiny, these crystals, she could fish these up and we could collect a diffraction pattern at a synchrotron and solve the X-ray structure. So this is a structure of a nut factor receptor of the NFR5 type. So it is really built out of 3 lyse-M domains. So there's one up here, a second lyse-M domain, and a third lyse-M domain. And they're kept together by 3 conserved disulfide bridges. So they are really in this clover leaf orientation. So from these atomic coordinates, we could calculate the electrostatic surface potential of the receptor. And what we realized that very close to the ligand binding site in M2, we find this very characteristic hydrophobic patch. So we were wondering, and that fits very well with, we have a nut factor ligand and then this fatty acid chain that could be positioned here. So this is something that we observed, but we solved the structure without the nut factor in there. So we wanted to understand if this hydrophobic patch is actually important for the role of this receptor. So when we take the wild type receptor and do binding experiments, we can see a certain binding kinetics. And then when we mutate the sites, so there was 2 surface exposed leucines, if we mutate these into aspartates that would add a negative charge and purify this protein, we could see that the binding kinetic changed quite dramatically. So there's lower binding, and when we quantify this, there's sort of much faster binding down here than here. So this wild type receptor is able to bind the ligand and maintain the complex or bound complex for longer, while this, when we mutate this site, loses the ligand. So this is, of course, sort of saying that, okay, this hydrophobic patch is important for the kinetic of binding, but we didn't know if this was important for the function. So to do that, we did complementations, and actually not in Lotus, but this is in Medicago, in a mutant called NFP. So this is the nut factor mutant. And when we complement it with a receptor that has these 2 mutations, we lose the symbiotic signaling. So it really says that the hydrophobic patch is important for binding the ligand and maintaining it for long enough for signaling to occur. And we call this kinetic proofreading, so that the plant can decide if it is a correct bacteria, correct nut factor, based on the binding to the ligand. So of course it's detrimental for the plant, right? It's really a key factor that the plant needs to react to this nut factor at very low concentrations, but also with it can't really activate this pathway in response to a ligand that's not correct. So because that could be a pathogenic, or that could be a incompatible symbiont, but it binds the nut factor specifically, and it proofreads the ligand by maintaining it bound to the structure for long. So we really think that kinetic proofreading is one of the key innovations that happened in NFR5 and that led to this transition from AM to root nodule symbiosis. So this was on one side of the receptor. There's maybe I go back a little bit. So we thought, okay, if this hydrophobic patch is really important, we might also be able to predict it, to predict that which other nut factor receptors are out there. So we did homology model of all the lotus receptors and found out that they are very different in the ligand binding areas. So some have very negative charges. We would think they are incompatible with nut factor binding. But we found this receptor, Lys11 in lotus, which has the characteristic hydrophobic patch. So predicting wise, we would think that this receptor would be able to bind nut factor. So this is all modeling. There's no experimental work behind this, but to prove it, we expressed the protein, crystallized it, and solved the structure and computed the electrostatic surface potential. And what we could see is that indeed it has a hydrophobic patch, exactly where we believe it should have. So we think from this that Lys11 would be a nut factor receptor. When we do binding experiments, we can see indeed it binds nut factor, but it also binds chitin. So it lacks the selectivity or the stringency. And if you remember before with this binding kinetic, the binding kinetics here are completely different. So they're super fast on rates and off rates. So we don't have any kinetic proofreading here. It's just binding and then release. And when we put it in plants, we can see that Lys11 is not able to complement the receptor. And we really believe that that's because it lacks proofreading. So then we thought, okay, can we then engineer proofreading? If this was a key innovation, can we then change it and to support symbiosis? So we identified 6 residues around the ligand binding sites, and we mutated these and purified the chimera. And what we could see is that when we have this engineered version, we get specificity. So it doesn't bind chitin anymore, but it binds nut factor. So there's specificity. And the binding kinetics somehow changed. So the retention time of the ligand to the receptor is much longer. And when we put this into plants, this chimera, it supports symbiosis. So it really shows that, okay, we can engineer kinetic proofreading, and kinetic proofreading is important for the function and symbiotic function. All right, so I think I was here before. So this was on the NFR5 side, that the LSM2 is important for kinetic proofreading and symbiotic function. But we also wanted to know if there was any innovations here in the NFR1 receptor. So to understand how NFR1 evolved, we compared it to its closest homologue, which is a chitin receptor in immunity in lotus called Circ6. So the chitin receptor Circ6, it looks very, very similar, both in sequence and on structure. It binds not nut factor, but an undecorated nut factor. So in acetylglucosamine chitins. And we're trying to compare these two receptors to understand what really differentiate them and what was important in NFR1 for evolving symbiotic and stringent LCO perception. So we solved the structure of the immunity receptor, Circ6. I'm showing just the cartoon backbone of this. And then later, a couple of years ago, we solved the crystal structure of the NFR1 type of receptor. This one is from Medicago, so it's called LUC3. And then we superimposed those two structures. And what we saw is, yes, they are indeed extremely similar on the overall fold, but there are also small differences, very small differences in this first lysem domain. So they differentiate a little bit in the first lysem domain. And doing a lot of complementation studies, we could show that whenever we do manipulate or we make these chimeric receptors, whenever we change the first lysem domain or put in a bulky residue in the first lysem domain, we abolish the symbiotic function. So here we are looking at nodules per plant, and then we put in complement with these different versions. And we can mutate the second lysem domain, the third lysem domain, but whenever we change anything in this first lysem domain, we change the symbiotic function. And we narrowed it down. So we found 2 regions, 2 small sequence regions in the first lysem domain that we call region 2 and 4 that have the whole function. So whenever we change anything in these 2 regions, we change the symbiotic function. So these two regions, we map them again back to the structure to understand where are they actually positioned on the receptor. And it turns out that these are the two small regions that actually contact the ligand bindings sites. So this is in the first lysm domain in a chitin receptor. And these two regions, 2 and 4, in a chitin receptor, they basically surround the chitin molecule. And in a chitin receptor, when we compare it among all the chitin receptors in legumes, we can see that these two regions are extremely conserved. And that makes a lot of sense because this molecule, chitin molecule, is not varying at all. All plants have to recognize this, and legumes have evolved a very conserved way to recognize this danger signal chitin. But when we compare these two regions to the nut factor receptor, what we see is that they're very variable. Just these two regions changed a lot because nut factors, they change. Different bacteria has a different nut factor, so you can't have one way to recognize all, but you really need some variability here. So this was a key innovation to really show that we think we understand this enough, because we still haven't crystallized it with the right ligand. We thought, okay, can we then engineer it a little bit again? So we took the chitin receptor, expressed the ectodomain in insect cells, and did binding experiments with a nut factor. And it turns out the chitin receptor, immunity receptor, does not bind nut factor. It doesn't recognize it at all. It recognized chitin with this very conserved mode. But when we took these two small regions from a nut factor receptor and put them onto this receptor, so made a chimera, then we engineered and regained nut factor binding. So it really shows that these two regions are key innovations in this NFR1 type of receptor. And when we put it onto a chitin receptor, we can regain binding. So this is only in the ectodomain. If we couple this to the kinase of the symbiotic receptor NFR1, it actually supports symbiosis. So we engineer specific nut factor binding, and that's enough to start symbiosis. Okay, so then the question for us was always, we knew that there were two receptors, NFR1 and 5, that were absolutely essential for the signaling pathway, but we didn't know how they potentially work together. And this is, of course, difficult to understand in a living plant. How do one receptor possibly work with another co-receptor, and how will these work in bigger complexes? So this is where I go back to sort of my background in the nuclear pore field from my work at MIT. So here we used a lot of the immune system from llamas, because they have a very simplified immune system where we can make ultra small antibodies. So we immunized the llama with NFR5, the kinase of NFR5, and then raised this ultra small antibody against NFR5. And then we fused it to NFR1. So we have a receptor with a small antibody that recognizes its possible co-receptor. And then in theory, so it took me really a long time to get funding for this, because people thought it was a little bit of a too simple and maybe also a stupid idea. But in the end, we got funding for it. And the idea was that we would be able to control association between two receptors, a possible receptor and a co-receptor, and then start to examine these in living plants. So when we do this and we have NFR1 and 5 together and we don't add any rhizobia, we don't get any nodules. So the system is dead. But if we have the two receptors where they are controlled their association by this nanobody, we actually get nodules. So we can start organogenesis by bringing the two receptors together. We don't need the bacteria there, and the nodules on lotus looks very similar to the wild type case. So we're really happy about that, because it really shows us that what's probably happening when the nut factor comes in is that NFR1 recognizes the nut factor and NFR5 recognizes the nut factor, and they form a larger complex that are important for signaling. So we really believe that NFR1 and 5 is the core signaling complex driving symbiosis. Then we were interested in how does the signal actually propagate from this. So if we need the nut factor to bring the two receptors together, how do we start the signaling? So we could show this by, by nanobodies. We put them together, we get organogenesis, so we start signaling. But we also knew that there was a third receptor, not a lysem receptor, but a third receptor called SUMAK that were somehow involved in this process, but we didn't know when. So we did the same experiment in the triple mutant, bringing NFR1 and 5 together, but lacking SUMAK. And in this case, we don't get nodulation. So it really shows that we need SUMAK somehow downstream to activate. And there's no way we can sort of overrule this. So we can't just bring NFR1 to SUMAK, or we can't bring NFR5 to SUMAK and activate the signaling. We need to bring NFR1 and 5 together first, and then we need SUMAK somehow downstream. So what we really needed to understand now is how do we activate SUMAK? So being structural biologists, we are a little bit sort of biased towards going for structures. So we thought, okay, if we need the SUMAK, let's find out what we actually need there. So Melita, a student in the lab, she purified the intracellular domain of SUMAK and crystallized it, and we solved the crystal structure. So this is shown here with this long extensive tail in the C-terminus. And we could show that, it's a pure protein, and it's able to autophosphorylate itself. So it's an active kinase that can autophosphorylate itself. We mapped all these phosphorylation sites. So this is the work by Nikolai. And then we really saw that there's some phosphorylations here, but there are massive phosphorylations in the C-terminal of SUMAK. So we are trying to understand which of these were important. So we did this, again, going back to a lotus plant that lacked SUMAK and then complemented it with a wild type, and we're looking at nodules here. So we can complement the mutant. Then we thought, okay, if we just cleave off this whole C-terminus where many of these patient sites are, what actually happens? And then we can't come. So it really shows that there's something in the C-terminus of SUMAK that are essential for signaling. So it could be in this most distal tail. So we chopped off that, but we can do that fine. SUMAK is happy, at least in root nodule symbiosis. So that means that it's probably these 4 sites here. We mutate these to alanines, so they're serines here, and we mutate them to serines, to alanines, and when we lose nodulation. So we really lose everything if these sites can't be phosphorylated. There's no additional effect by mutating these sites. So it really seems that it's the sites in what we call the alpha I motif. So this long helix in the kinase is the I helix in the kinase. So we call it the alpha I motif because it sits here on the border between the alpha I helix and this region. We don't know yet, or at least in this study, we don't know yet if it's single residues out of these 4 that has the whole phenotype, or if it's a combination. But we think it's a combination from these next follow-up experiments. All right. So then we thought, okay, if these phosphorylations are important, and if we mutate them into alanine, we create a version of a receptor that doesn't work. We thought, what happens if we put in a phosphomimicking version? Can we then start the program? And luckily, we have amino acids, an aspartate that looks very much like a serine that's phosphorylated. So if we made these 4 sites into aspartate, we actually would have a phosphorylated receptor or kinase. And when we do that, we activate the program. We don't need bacteria at all. So if we just put in these 4 mutations in SUMAK, we can drive the signal. And there's no additional effect by having phosphomimics and everything. So we really created a gain of function receptor where phosphorylation of the alpha I motif drives root nodule organogenesis. Okay, so going back to the overview. So we know NFR1 and 5 perceive nod factor, and then we know that phosphorylation of the alpha I motif drive root nodule symbiosis. But we were wondering, what is actually the role of NFR1 and 5, how does it do the signaling from CMYK once it's phosphorylated depend on NFR1 and 5? So we did the same experiment where we put in a gain of function receptor in a plant that lacked NFR1 and 5, and here we can actually drive symbiosis. So we don't need NFR1 and 5 once the alpha I motif is phosphorylated. What we don't know yet is exactly how the signal comes from the nut factor receptors to activate this. So this we're working on. But we can show that if we take a component out, a kinase that decodes calcium spiking called CKMK, then we don't get any nodulations even when we have a gain of function CMYK. So it really acts upstream of this and downstream of the nut factor receptors. Okay, so going back to this overview slide. So we were interested in understanding exactly what happened here that enabled this transition from AN symbiosis to root nodule symbiosis. And we know that stringent nut factor recognition, proofreading, and the variability in LZM1 is important for this. But why are we doing it? So we are trying to see if we at some point can recreate this. Of course, this is super ambitious, but maybe if it happened here, that we could actually recreate this trait somewhere else. So in monocots, in cereals. So the prediction would be that cereals, they have maintained the symbiotic signaling pathway, the old one, that still supports a vascular mycorrhizal symbiosis, but they haven't made this transition into root nodule symbiosis. But cereals, they do have AM symbiosis, so they should have these receptors that can activate symbiosis. They do actually respond to the same ligands. So you can see here in a comparison with calcium spiking that cereals do have these receptors that can activate the common symbiotic pathway that are shared between the two programs. But there's also some differences. So that means the next question we asked down here is that can we, using our knowledge from legumes, can we identify which receptors in cereals that are responsible for calcium spiking or the symbiotic signal? So we looked here now in barley, and we compared, we found barley receptors, LZM receptors that was closest to NFR1. We found this barley receptor called RLK4. So there is receptors that sort of on the phylogeny looks closer to a nut factor receptor of an NFR1 type. And the same was for an NFR5 type. We found this RLK10 to be quite close to this. And being structural biologists, this is only modeling, so we haven't solved the structures of the barley receptors, but here we compare the molecules, and we can see both in their ectodomains, the ligand binding domains, and in the intracellular part of them, they're quite similar. So we believe that this is the sort of the old complex that can activate symbiosis. But we wanted to prove that. So we took these two receptors, RK4 and 10, and put them back into legumes to see if they could support symbiosis. And what we found was that they couldn't. So we think they can activate the common symbiotic pathway, but they're lacking something. They haven't gone through these key innovations that enable them to support root nodule symbiosis. But we thought, okay, what if they lack this stringency that nut factor recognition or LCO recognition that are actually essential? And we can overcome this by bringing them together with nanobodies. So we made a nanobody that had affinity for the co-receptor, and then with the idea that we could bring these two together. And when we do this, we can get nodules with the serial receptors. And the nodules looks very similar to the nodules we get from the native receptors. So we believe we have identified this old complex that can activate the common symbiotic pathway, and in cereals are important for AM symbiosis. And we believe that this receptor complex is what we need to try to engineer if we want to recreate this trait in cereals. Trying to figure out what I wanted, my point here. But basically, we are lacking something. So we know that we can't just take these serial receptors and put them in legumes to support symbiosis. And we think what they really lack is this stringency. So they need to be able to monitor the nut factor specifically and with the right stringency to support symbiosis. So this is very much what we're working on. I have one extra piece of data because I've shown a lot that there are probably some key innovations here in the extracellular domains, but we believe we also found some key new motifs or innovations in the intracellular domains. So we have now started to examine the intracellular domain of NFR1. We solved the crystal structure of NFR1 and found that there is this motif up here. We believed that before we solved the crystal structure, we actually believed that this was the juxtamembrane domain. So the domain here that connects the kinase to the transmembrane helices. And we thought that would be a flexible just linker that connected the two domains. But what we found from the structure was that it's actually an integral part of the kinase domain. And it's very, very conserved in the symbiotic NFR1 type of receptors and different to the chitin receptors. So there's something here that seems to have evolved in NFR1 that are important for symbiosis. And we found two residues. And if we mutate these, we change the symbiotic function. But maybe more sort of illustratively, if we take a chitin kinase that doesn't support symbiosis, but if we then, so this is Circ6, if we mutate it back into, so it looks like a symbiotic kinase, we can get a immunity kinase to support symbiosis. So we were quite thrilled when we saw this, and this is just changing two residues. And we did this with the barley receptors as well. So barley receptors doesn't support symbiosis, but when we change these two residues, we can get it to become symbiotic. So we really think that we found a innovation in the intracellular domain of NFR1 that we need to understand really to start this signaling pathway. So I think this was the last sort of data I have to show. I don't know how I am with time, or we have time for questions and so. So thank you very much.